So slight change of track. So for the second part of the talk, I'm going to describe some of the principles we use for investigating potentially problematic RCTs. All right, so just some context. So for this part of the talk, I'm mostly drawing on my experience in doing these kinds of investigations for journals and publishers over the last 4 years or so, I'd say. I was very lucky to receive some training by Stephen Evans. So he's a real expert in this, a really shrewd guy. He gave me some training in this. Although to be clear, if I say anything daft, that's my mistake, not Stephen's. All the cases I work on are confidential, so I can't talk about specific cases. And so all these examples are inspired by real cases, but I've changed details. So I've actually fabricated data for this part of the talk. All right. But to be clear, when we do these investigations, it involves a really thorough examination of everything, right? Manuscript, data, other resources like the registration entry, correspondence with the authors, etc. And it's really important to note that we're not really trying to prove misconduct when we do this. Our goal is much more modest. We're just trying to answer the question, could these data have arisen from a genuine RCT? Could they be authentic? But the conclusions are based on a holistic assessment of everything, not like a single statistical test or anything like that. I think that's a misconception. This usually takes, a day's work, at least for me, to go through a data set and everything. So it really is quite laborious, takes quite a lot of work. And just to be clear, right, I'm illustrating a few basic principles we might rely on here, certainly not being comprehensive, certainly not a tutorial. If you are a fabricator yourself watching this, okay, we're certainly not showing our full hand here, all right? So don't get too excited. So what do these investigations look like? Let's think about this. So usually what happens is some concerns are raised with a study on the basis of published manuscript or a submitted manuscript or what's in the trial registration entry. Just to be clear, right, I put a random paper up by some collaborators actually in the top left. I do not mean to imply at all that there are any concerns about that study, all right? But usually what happens if, those concerns look like, yep, they are sort of substantial concerns on the basis of there, we'll probably move to a second stage, which is a more detailed investigation, right? And there we're requesting additional documentation from the authors, trying to get hold of the underlying data set, and then performing some analysis of the data set as well. And for both of these stages, we're really interested in the question of how, right? So how can we do these things well? And sort of recognizing that there are these two stages, we've kind of planned for two versions of the tool, right? So we'll have Inspect SR, which we can apply when we've just got the stuff available at stage one. And if we can get hold of the IPD, or when the IPD is available, an extension to that, Inspect IPD. And let's just remember our goal here. So we really want to make sure that this fake data can't influence patient care, but we also want to avoid unintentionally removing genuine data too, all right? Because genuine data is precious, valuable. People have given up their time to do this research. We don't want to remove it incorrectly. So for the last part of this talk, I'm just going to focus on some of the IPD methods to tell you a bit more about those. So let's think a little bit about what happens in an RCT. How are people recruited and allocated to treatments in an RCT? Well, what happens is that potential participants will present over time, okay, and they're going to have their baseline measurements taken before randomization. So I've got my queue of people presenting to be randomized in the trial, and they're going to have their baseline measurements assessed. And then anyone who's eligible is going to be sequentially allocated to study arms in a random sequence, okay? So my first participant's in arm 1, so is my second participant. My third participant's in arm 2, so is my fourth participant, all right? Now, there are some hopefully quite well-known consequences of that. So first of all, we don't expect to see any systematic differences between the groups in terms of their baseline characteristics. But perhaps a less appreciated point is that any patterns in baseline characteristics over time should be present in both groups. So for example, say there was some sort of change in the measurement process here. That could be, for example, a problem with a measurement device, or maybe a new investigator becomes involved and starts targeting a different type of patient. Well, the point is that if there is some sort of change like this should start to be present in patients allocated to both arms of the trial around this time, okay? So we can look for things like this, all right? So let me give you an example. This is an example of some genuine data, actually. And what I'm showing you is a three-arm RCT with blue vertical lines dividing the data into those three groups, A, B, and C. And all we've done is we've plotted a baseline measurement in randomization order within each one of those groups, okay? And if we look at group A, first of all, you might sort of say like, is there something weird going on there? Suddenly we get a few sort of higher measurements. Maybe the variance is a bit greater there. That looks a bit odd. Well, the important thing is that we see this happen in patients randomized around this time in the other groups too. And that is what you'd expect to see under genuine randomization if there were some sort of problem like this. So that wouldn't be a cause for concern at all. Let's look at this example. So this is a two-arm RCT this time, but same idea, a vertical line dividing into two groups. And in the group on the right, you sort of say like, there's a period here where the data looks really weird. We've got much larger values, different distribution here. Well, we don't see anything like that in patients allocated around the same time in group A, okay, which we would do under a genuine RCT for a baseline measurement. So this doesn't appear to be compatible with a genuine RCT. These kinds of plots can be useful for revealing all kinds of problems too. So if you want to just take 10 seconds or so to have a look at this plot, can you see any apparent problems here? Again, just a baseline variable in randomization order within group. Do you see any issues? Well, perhaps you notice that there appears to just be repetition. Okay, so this is a case where somebody's taken a sequence of values and just copied and pasted them, perhaps editing them slightly over and over again in the spreadsheet. And we can also look at outcome variables too. So not just baseline variables, but also outcomes. They're a little bit different because outcomes are influenced by treatments, okay? So we do expect to see differences if there is some sort of effect of the treatment. However, these kinds of plots can still be quite useful, all right? So what I'm showing you here is a categorical outcome variable, which takes three values, 1, 2, or 3. Again, just plotted in randomization order in treatment arm. Have a look. Do you see anything unusual about this one? Does anything look odd? Just give you a few seconds. Well, you may notice here that the outcome variable basically occurs in long runs of the same value. So in the data set, we have a long run where everybody gets an outcome value of 1, then everybody gets an outcome of 2, then 1, then 3. This isn't what happens, okay, in real data, all right? This does not appear to be authentic. Very briefly, another thing we look at is we look at correlation across rows in the data set. So what's the idea here? So the basic idea is that you don't really expect to see substantial correlation between the baseline values of successive participants in a trial. So on the right here, I've shown you the first 15 rows from a data set, and we've got the duration of infertility for those people, which is a baseline measurement. And so one participant's duration of infertility shouldn't be related to the next person's duration of infertility or to the person after that, etc. However, you do expect to see some sort of correlation across the rows if someone has sat down and just typed some numbers in a column into a spreadsheet, because people are quite poor random number generators, okay? But even if there is some autocorrelation, we call it, we certainly don't expect this to differ between randomized groups. And we can look at this as well, all right? So this is what's called an autocorrelation plot for treatment and control groups. What is this showing you? So on the y-axis, you've got the degree of correlation, if you like. And as we move along the x-axis, we've got increasing lags. And we're showing you the plot of correlation between the duration of infertility values 1 row apart, 2 rows apart, 3 rows apart, etc. And what we can see is, in the treatment group, there is this quite substantial correlation between successive rows, which sort of decays as we get further apart, rows further apart. But that isn't present at all in the control group, okay? The control group looks like genuine data there. There is no serial correlation. And what I'd say for that one is the treatment group correlation is quite suspicious But the fact that there's this big difference between groups in this baseline measurement autocorrelation is even more so. And finally, I'll just mention another thing we can look at. So we tend to look at the relationships between the variables in the data set. Are they as expected? Okay. And that's quite hard to fake unless you do know what you're doing, I suppose. But this kind of thing really does require contextual knowledge. So it came up in our answer to the question before, but this is an example where you probably do need some contextual knowledge to understand what the relationships should be. And again, we know randomization really helps us here. We don't expect this multivariate distribution to differ between the randomized groups, and that's something else we can consider. Okay, so just to finish, so yeah, this was just showing you a few very basic checks here, right? And there's lots and lots of different approaches. I mean, I think I showed you that we had around 41 IPD checks that we were considering at the moment. Different approaches are going to be more or less appropriate for particular cases, all right? But the main point is that we understand quite a lot about RCTs. We understand quite a lot about what data should look like if it's arisen from a genuine RCT, and we can basically use that knowledge to assess whether data are compatible or incompatible with genuine RCT. But of course, we always need to think, could there be an explanation for this? Okay, and in these investigations, giving the authors a chance to explain any unusual patterns that we find. Sometimes when we're doing this, there's really clear evidence of fabrication. Other times, it's unclear whether you're looking at misconduct or just really poor conduct, all right? But arguably, it kind of doesn't matter, right? If we know the data are that unreliable, if we know that they couldn't be authentic for whatever reason, we're probably going to have some reservations about allowing those data to contribute to patient care. So just to close then, I just want to say thanks to the expert panel members on this project. And just to remind everyone, so we will need people to contribute, to input. So need people to participate in a Delphi. We're going to need people who are going to be willing to test the tool while undertaking a systematic review. As we heard in the questions, actually, that includes editors as well. We're looking for editors to include in that testing. But basically, if you've got any interest at all, just please contact me. You can get me on my email or my Twitter. So thank you very much. I'd be happy to take some more questions.